Hello. I apologize about the last video. Had some trouble with the sound. Anyway, we're back. We talked last time about the two dimensions that you can classify a study by. The first is census versus sample, and the conclusion there was that census versus sample is not nearly as important an issue as a person first thinks, and the reason is because, except in special cases, the thing to do is sample. And we talked about how if you try to take a census in a context, anything other than a special case context, then if you try to take a census, you're likely to end up with results that don't reflect the actual underlying population because you're getting incomplete results and they're going to end up biased. So we find ourselves looking at the second dimension today, observational study versus experiment. So again, when a person first approaches statistics and they think about what's the most important thing, this is not what strikes them as the important thing. And furthermore, to the extent that they think about it at all, observational study strikes them as being like sort of a fifth grade view of mathematics and science and computer science. So it strikes them as being kind of that what you're supposed to do. You're supposed to observe nature. Experiment sounds suspicious. The observer affects the data set. And that sounds like you're reaching in there and tweaking with the numbers, which sounds at least a little bit peculiar. I hope to convince you today that that's exactly backwards. That in fact, census versus sample is not the important question. The important question you should ask yourself when you're looking at a study is it observational study versus experiment? And to the extent that it's an experiment, you're in general, usually, you're saying, oh, okay, that's great, that's what we're looking for. Okay? So let's talk about the observational study versus experiment case. Let me click forward to that. There we go. So observational study is where the investigator simply observes the variation of interest. Experiment is where the investigator imposes treatment on some individuals and withholds it from others. There are many complicated cases, and we'll talk about some of them today. But trying to present a very first impression, the investigator imposes treatment on some individuals and withholds it from others. Those are called the treatment group and the control group. The investigator acts so that, as much as possible, and again, this is a first take, acts so that as much as possible the two groups on average differ only on whether the treatment is applied. Various strategies, and we'll talk about them today, various strategies are employed to keep all the other differences constant. The most important strategy is randomization. We talked some about randomization already. So I'm going to talk first about observational studies, and as we move from the top of the slide to the bottom of the slide, I hope that a person will see that we go from observational studies that are good, are admirable, down to increasingly problematic. So the first observational study is Jane Goodall in 1962 or whatever it was. Jane Goodall goes out to a tree that has a lot of fruit and just sits there and waits. You think, imagine the mosquitoes. It just sits there and waits, and the chimps show up, and at first they're like, whoa, what is this? But eventually they get used to her, and at that point she starts writing down what do they eat, how do they act, who shows up on Tuesday, who also showed up on Thursday, kind of stuff. Okay? Everybody thinks that the best thing you can do in that circumstance is to simply observe the chimps. That's right. Going to a zoo and watching them proves to be very misleading. The right thing to do is to sit in the wild and observe them. That's right. That's good. Okay, but as we move down through the slide, we'll see increasingly problematic things. And the next example is a good one. So the effect of unemployment on corporate profitability. So you go to the you go to the library, maybe you look it up in Morningstar, you find a table comparing the national unemployment rate to the price earnings ratio for companies in the Fortune 200 for the past decade. Okay, so a person says, well, I couldn't really do much besides observe this. What am I supposed to do? Create my own economy? So, of course, I just have to write down the numbers that come. Yes, that's true. But certainly a person who reads this study is going to say to themselves, well, perhaps the last 10 years were different. Perhaps if you had a longer period, 30 years, 50 years, post-war kind of stuff. And so a person is starting to see that there are all kinds of questions because all you did was write down the numbers. The objection that all I could do was write down the numbers is very sensible, but nonetheless, it leaves open questions. Next example, we compare the 100-meter dash times of students who take a daily multivitamin with the times of students who don't. And again, we're getting to increasingly suspicious studies here. Pardon me. So, what do you think? I mean, my first thought, if I was listening to that talk, would be that students who take a daily multivitamin, how many college students you know go to the trouble of taking a daily multivitamin. That students who take a daily multivitamin are likely to be very different than students who do not. Just to name an example, I bet you that among the people who take a daily multivitamin, I bet you very few of them are smokers. Just to name an example, I bet you they have on average a lower weight, just kind of stuff. And maybe it's the smoking, maybe it's the average weight, maybe it's the weight. In other words, it isn't the multivitamin. The study that you're doing in no way proves that it's the multivitamin. Okay, so next. I like this example because it's a lesson sort of for our time. Everybody's got databases, everybody's got piles of data, and there's a sense of, well, we have this data, we should look at it. And I agree with that to some extent, but a person has to be aware of the limitations of what you're looking at. So we wonder whether an older mother is in part the cause of troubled pregnancies. So we work at a hospital, we open up our database, we make a table comparing the age of the mother to the percentage of pregnancies that are either routine or mildly troubled or quite troubled. And you have some criteria for those three conditions. I mean, maybe, the routine would be the number of doctor visits is below this number. Mildly troubled is the number of doctor visits is between this and that sort of stuff. Okay. So again, if I was reading that study, one of the things I would think, well, what is this hospital? I mean, if you work at St. Albans Hospital, nothing wrong with St. Albans Hospital, but if you work at St. Albans Hospital and you are looking at, say, a 35-year-old mother, well, 35-year-old mothers have more money than 20-year-old mothers. So if you're a 35-year-old mother, are you going down if you're having a trouble pregnancy, are you going down to Burlington? Are you going to Dartmouth? Or what do you, so in other words, is the data not what it seems to be? Because all you did was open up the database and look at the numbers that you got. And then finally, the classic example here: doctor goes somewhere and learns how to do some new procedure for blocked arteries, and they bring in patients, the 417 patients in the past year or whatever it is, and the patients, they say to the patient, "Here's the new treatment, here's the old treatment. What do you want to do?" And the patient chooses one or the other, and the doctor writes a paper that reports. Okay, the problem with that is that the patient chose one or the other. If you're an 80-year-old and the doctor says to you, okay, so we can do this surgery where we snake a wire up through your veins and then or we can go with the current treatment, which is this course of drugs that has worked for many years, what are you going to choose? But if you're a 30-year-old, what are you going to choose? I mean, if I'm a 30-year-old, I say, go ahead and get that stuff out of there, kind of stuff. So that is to say, if patients self-select themselves into treatment versus control group, all of a sudden many, many questions arise that didn't arise before. You've convinced, your study is far, far less convincing. I want to compare that to experiments. And of course, I'm taking, this is a first take on the idea, so we're taking a somewhat extreme idea, but nonetheless, that is to say, many studies are a blend of observational study versus experiment. But nonetheless, I want to give a person the impression that observational studies often leave questions in the reader's mind, and experiment often answer the questions. Okay, so here's an example. Polio hit the U.S. in 1916 and until the 1950s killed many people. How many people you know had polio? Polio is at this point largely a solved problem. Scientists could see in a test tube that the vaccine developed by Jonas Salk caused the production of antibodies, but does it prevent disease? I mean, it works on mice and it works on people, are two very, very different things. If you simply gave the treatment to every child in the country, then a drop in the disease rate might not be convincing. For example, the 1953 rate was half of the 1952 rate, just because for some had to do with the weather, who knows? So in one study, researchers split the children into two groups. I remember this kind of thing when I was a kid. There'd be some announcement on the classroom speaker. Everybody would line up. We'd all roll up our sleeves. We'd all march down to the nurse's office. Everybody gets a shot. Half the people in the class got vaccine. Half the people in the class got salt water. Nobody knew what was in the tube that they got. You rub your arm on the way back to the class. You sit down, and now you start in class again, and it's all it's all done. When it's all said and done here, the group that got the vaccine and the group that got the salt water had very different statistics. If somebody in the talk about this raises a hand and says, "Well, could it be that because there were more boys in the vaccine group than in the saltwater group?" The answer is no. We randomized it. We flipped 50-50, vaccine versus salt water. So it's going to be 50-50 boys versus girls. Could it be the age of the children? Because older children, no, we flipped a coin, 50-50. So older children half the time, not as old children half the time. It's all because we flipped a coin, we got rid of that factor. Could it be East Coast, West Coast? No, no, all those factors are gone. All those questions are answered. Because we have a randomized treatment and a randomized control group, all those questions are answered, or at least the great majority of those questions are answered. The vaccine had a much lower rate of polio. The vaccine prevents polio. You know the answer. And please, I get it. There are all kinds of issues that occur to a person. You say, a mother comes to you and says, please, I lost 3 boys to polio. Could you please put my son in the treatment group? But these things are very hard. I'm not saying that they're very, but look at the data. It answers the question. How many people you know have had polio? The question is answered. Many observational studies have found that people who get extra vitamins because they eat 5 or more servings of fresh fruit and vegetable every day, especially broccoli, have much lower rates from colon cancer and lung cancer. Lots of studies, lots of observational studies found that. On the basis of that evidence, researchers did a large randomized controlled experiment. A treatment group got extra vitamins, the control group ate their usual diet. No difference. So again, because it's randomized, it settles the question. It turns out that people who eat 5 or more servings of fresh fruits and vegetables every day are very different than people who fall in the other category. They, for example, I bet you that they smoke at much lower rates. I bet you they exercise at much lower rates, etc. It isn't the fresh fruits and vegetables necessarily. It is something else. I don't know what, but it isn't the fresh fruits and vegetables. In particular, it's not the extra vitamins. The two groups are very different. And unless you do a randomized controlled experiment, you haven't convinced anybody. People will remember this kind of study and say, okay, but when the next study comes along that didn't do a randomized controlled experiment, I got questions. Another example. I don't want to give you hundreds of examples, but I want to give you enough to give you the sense that I could give you hundreds of examples. Okay, so here's an example from the Burlington Free Press back when it used to be a newspaper. So if you're one of the myriad toiling American workers arguing that you can't take a summer vacation, you're too busy, here's one reason to rethink your decision. It could save your life. A researcher found that middle-aged women who took vacations very infrequently, defined as once every 6 years or less, had 8 times the risk of having a heart attack or of dying of heart disease. And I clipped a lot of the article, but basically the article said hard-driving lawyer types, career people who don't have time to take a vacation, that they're dying of heart attacks. But when I read the article, and please, this isn't the original study, so perhaps the newspaper didn't do right by the person who did the study. That's perfectly fair. But when I read this article, what occurs to me, who do I know who takes less than 1 vacation in 6 years? Maybe a hard-driving lawyer type, maybe. But I also think of middle-aged women, the woman who comes in and cleans the room after I leave the hotel. I mean, this kind of stuff. They're not taking vacations. So I'm thinking of them, and I'm thinking of the people who work 3 jobs at convenience stores. They're not taking vacations. I'm thinking I'm thinking here that because this is an observational study, it leaves a lot of questions in my mind about what's the underlying reason. Is it from being a type A person, or is it because when you work 3 jobs at different convenience stores, you're stressed, you're smoking, you're eating not so good, sort of stuff. Sleeping not so good, sort of stuff. Okay, again, a couple of examples, not too many I hope, but enough so that a person will remember, walking out of this video, will say to themselves, okay, there's overwhelming evidence for experiment versus observational study. The disease pellagra, first observed in Europe in the 1700s, especially people of northern Italy, spread in the 1800s, southwestern France, parts of the Turkish Empire, Egypt, South Africa, prevalent in the south of the U.S. Seemed to hit some villages more than others, some households more than others. One blood-sucking fly had roughly the same geographical range as the area of greatest disease, and furthermore, the fly was most active in the spring when the disease was most prevalent. So most epidemiologists figured, on the basis of this observational evidence, figured that this was just another insect-borne disease. But in 1914, the American epidemiologist Joseph Goldberger showed, in a combination of studies and observational studies and experiments, that pellagra was not not about blood-sucking flies. It was caused by bad diet. Milk, meat, and eggs contain a nutritional component that prevents pellagra, but corn contains very little of that component. Poor people ate corn. Since the 1940s, most of the flour that's sold in the U.S. is enriched with a vitamin that prevents the disease. They used to call it pellagra preventive factor, but the marketing people didn't think it was a great idea to have a big sign on the front of the bag that says contain PP. So instead they changed the name to niacin. That's what niacin is. So now the bag of flour will say on the front contains niacin. That's to prevent you from getting this disease. How many people you know have this disease? Again, just a couple of examples, but enough, I hope, to leave a person with the sense that there are thousands of examples. Okay, so researchers in several different countries observed that cervical cancer, quite rare among Jewish women and Muslim women. In the 1950s, several investigators announced that they were satisfied that the circumcision of the husbands was the protective factor, because they're men. But there are other differences between Jews and Muslims and other groups besides circumcision. We now know that cervical cancer is a sexually transmitted disease. You can see when you take the cancer cells, you can see the DNA of the papillomavirus in the cancer cells. I mean, that's overwhelming evidence. Early researchers didn't pick up on it, in part because cancer takes long to develop. It isn't about who your husband was in the 1950s. It is instead about whether you were sexually active in the 1930s. And Jewish women and Muslim women tended to be less sexually active, and so they got the disease less in the 1950s. So this observational evidence seemed very strong indeed, but it proved to be wrong. The conclusion. Observational study versus experiment is the crucial dimension on which to judge a study. If you find yourself in your life saying, my dad has to have this procedure, I'm going to go on the internet and see what I can find out about it, read a bunch of studies, some of them are observational studies, a few of them are experiments, you should think to yourself, the observational studies historically have often been wrong. So that's the crucial dimension on which to judge a study. You want studies to be, to the extent possible, experiments. Experiments give the right answers, often, while all too often, observational studies give the wrong answers. In an experiment, the investigator institutes control and treatment groups, adopts additional techniques where needed, we'll talk about that throughout the course, but also just later on in this video, and arranges a study so that, to the extent possible, the groups on average differ only in whether or not the treatment was applied. Okay, so we have some vocabulary that I want to use and introduce, and of course the concepts that go with the vocabulary. So two variables are associated if they vary together. So you think, for example, that smoking is associated, are you a smoker, yes or no, is associated with your chance of getting lung cancer or with your chance of getting heart disease. So two variables are associated if they vary together. The association is about a cause. The association is causal if changing one variable influences the other. If so, then the first one is the explanatory variable. Smoking is the explanatory variable. The second is the response variable. Cancer is the response to smoking. Since observational studies don't control any variables, the results can only be associations. It's hard to infer a cause from an observational study. If the variables are controlled, as in an experiment, then you have a stronger argument for cause. So a very typical example, we want to test a new blood pressure pill reduces strokes better than the standard treatment, so we split the subjects into two groups at random. For the same reason why we split the polio children, the children in the polio test at random. One group gets the new pill, one group gets the old pill. At the end of 10 years, the treatment group has fewer strokes. So we've got a very strong argument that the pill works. So here the categorical variable pill type is associated with the quantitative variable chance of a stroke. Because this is an experiment, we have strong evidence that changing to the new pill results in a lower chance of stroke. If we had just watched people who chose whether to take the new pill or not, there'd be all kinds of questions. A confounding variable, often called the lurking variable, is one that's associated with both the explanatory and response variables. And the classic example I like the best is in elementary school children, if you draw a graph, and along the x-axis you have their height, and along the y-axis you have their vocabulary score. What you'll find is a pretty good match. Taller elementary school students have larger vocabularies. Okay, but height is not somehow cause you to have higher vocabulary. The explanation, of course, is that taller elementary school children are older, and older have higher vocabulary. The lurking variable there, the confounding variable, is age. Okay, so back at that Holiday Inn in Kansas City, professional researchers, the people in the seats there, professional researchers know these stories and the concerns that they bring up. If you're reporting on a study of yours and you could have done things to make your study stronger, but you didn't, that'll hurt your credibility. An investigator, before they set out to do a study, has to anticipate questions and arrange their study to cover the questions that people are going to ask. So as we move forward now through the rest of the video, I'm going to talk about some of the ways to cover some of the issues that are involved. Control group. First question that people often ask, first thing that people in this class often think to themselves, people who are learning about studies for the first time, why do you need a control group? So for example, why not just give everybody the treatment and see if it works? So I'm going to present to you, I could present hundreds, I'm going to present to you just a couple studies that did not have a control group and came to the wrong conclusion. I mean, what more argument do you need? Okay, so cirrhosis of the liver, a patient can hemorrhage. One treatment is to redirect the blood through a shunt. It gives the blood a place to go. This summarizes the studies on the effect of the treatment. There are 3 types of studies. Studies that had no controls, everybody got the operation. The controls were there, but they were not randomized. You brought in patients and you said to them, do you want to get the new surgery or do you want to continue with the old process? And finally, the tough one, randomized control. People come in the room and you say to them, you basically flip a coin. You get the new treatment or you don't. That's a very hard thing to do, but that's what randomized control is. Okay, so no control. If you look at the studies that had no control, that just gave everybody the surgery, then what you see is that most of the studies are strongly enthusiastic, think that the surgery works great. If you have a control group but it's not randomized, patients choose themselves into the control group or not, then again, you find that most of the studies are strongly enthusiastic. So if you're looking around on the internet, there's been this recommendation for your uncle or something, you're looking around the internet trying to find out information, and you're reading these kind of studies, no control, or these kind of studies, control group but not randomized, you're saying to yourself, okay, so that operation seems like it works. But when you go down to the gold standard, when you go down to the best way to determine the facts, it's a very hard thing to do, but it's the best way to determine the facts. What you find is the exact opposite. That if you institute randomized controls, the studies find that the surgery does not work. It doesn't work. These observational methods or more observational methods came to the opposite conclusion than the ones that had a randomized control group. If you're in that Holiday Inn in Kansas City and people raise their hand and said, why don't you have a randomized control group? You better have a good answer, because those people are thinking to themselves, I can name studies where came to the wrong conclusion because they lacked a randomized control group. Whoops. Oh, I misremembered what happened next. I'm sorry. Here's a question. Suppose somebody says to you, okay, why do you need a control group? Why don't you just compare to the numbers that you already have from historical studies? So, for example, here's that same shunt procedure. We know that 45% of people who do not get the shunt procedure, we've been studying cirrhosis of the liver for many years, we know that 45% of those people will not survive 3 years. Why can't we just use that number? Okay, and the answer is that right here. When you look at those studies that used historical controls, they found that the 3-year survival rate was 60% with the treatment and 45% without. Now remember, they didn't have a control group. They just relied on the numbers from the last decade. When you looked at those studies that had randomized control group, they found that 3 years from now, 60% of the people who got the surgery are alive, and 60% of the people who didn't get the surgery are alive. The surgery doesn't change the 3-year survival rate. Randomized controls tells you something that historical controls does not. Historical controls would lead you to get the surgery. Randomized controls would lead you to not. Coronary bypass surgery. Surely everybody's heard of coronary bypass surgery. Does it help people live longer? So this is exactly the same kind of stuff. If you use historical control If you look at the studies that have relied on historical control, they looked at the 3-year survival rate of people who did not get coronary bypass surgery sort of 20 years ago, and they looked at the 3-year survival rate of people who get coronary bypass surgery today. It's a big difference. You say to yourself, I would go for the surgery. Here, if you do it right, if you do randomized contemporaneous controls, you do your study by today having a treatment group and today having a control group with all the options of today's other treatments, medicines, whatever things you're getting today. It's not as clear case, is it? That the people who get coronary bypass surgery have a larger 3-year survival rate, but it's not nearly as impressive as the other way around. So what we're seeing so far here is that the people in the seats in that Holiday Inn Kansas City are saying to themselves, you gotta have a randomized contemporaneous control group, because I can name many studies that did not. Came to the wrong conclusion. I mean, you want the right conclusion? Why else are you in the business? Okay, what else do you gotta have? So I imagine that everybody has heard of the placebo, so let's talk about it. So you want to see if taking vitamin A helps students to learn. So you select a group of students, divide them at random into a treatment and control group. That part sounds good. You give the treatment group vitamin A pill to take every day. You find that there's a positive effect. When you show up at the professional meeting, what question are people in seats going to ask? Well, exactly what they're going to ask is they're going to say, but there's another difference between treatment and control group besides the vitamin A, and that is the treatment group took a pill. Could that have made a difference? For example, maybe people who are taking the pill are saying to themselves, well, I'm getting this treatment. I bet I can study longer. A placebo is an inactive substance of preparation used as a control in the experiment. So that is to say, the person in this first study, the vitamin A study, should have been giving the control group also a pill. Just a pill that doesn't do anything. So if you want to test a treatment to lower blood pressure, you split the subjects into treatment and control. If you only give a pill to the treatment group, then there's a difference between two groups. Somebody could wonder if the control group, for example, people who are not getting the pill, made a point of exercising more, or made a point of taking special vitamins, or whatever kind of stuff they're doing. Maybe the treatment group felt free to have a few extra Cheetos. That is, there's differences between the treatment and control group besides just the treatment. So you got to give them both a pill. One of them has the medicine in it. One of them contains a medically inactive pill, starch or something, just that sort of thing. Another example, researcher testing the effect of a new cholesterol drug breaks the subject at random into treatment and control group. Treatment group gets a new drug. The others get a similar looking pill with an inactive contents, and that's the right way to do it. People's response to placebo is very funny business. People's response to a placebo can be positive or negative. For example, in one study, people were given a placebo and told it was a stimulant. After they took the pill, their pulse rate sped up, their blood pressure increased, their reaction speeds increased. You can measure these things. When people were given the same exact pill and told it was to help them sleep, they experienced the opposite effect. So this is not in people's head. It actually happens that their blood pressure goes up. Responses are measurable. Furthermore, although nobody knows why, the placebo effect is getting stronger over time. So if you go to that Holiday Inn in Kansas City and you're given a talk about a new drug, people will say, have you demonstrated that the new drug's effect is larger than the effect of a placebo alone? What else? What other things are there people in that Holiday Inn in Kansas City? What other things have they learned about control groups, treatment groups? So blinding. If the subjects know that they're receiving treatment or know that they're receiving the placebo, then there'll be a difference between the two groups besides the treatment. So if we split the participants in a weight loss study into treatment and control group, and we have the treatment group come in on Tuesday and the control group comes in on Wednesday, it's not too long before the two groups notice that one of the two groups is losing weight and the other group is not, and they figure out which one is the treatment group. Experiment is called blind if information about the test is withheld from the participants to reduce the bias. The bias can very well be unintentional or unconscious, and if you have a protocol that includes blinding, that doesn't mean that you're saying to the subject, we know you people are going to cheat. Nothing of the kind is implied. You're just trying to get the best possible study. So if somebody raises a hand and says, did you do blinding? You say, yes, we did blinding. Double blinding. An experiment is double blind when the information is withheld from both the subjects and the tester. You can imagine, for example, you're in a hospital and some patients get the new drug, some patients get the old drug. Person comes around with a clipboard to evaluate. So this person is getting the new drug. I wonder if they look better than the people with the old drug, kind of stuff. Instead, the person who comes around with the clipboard should not know which of the two are they new drug or old drug. So to test the new cholesterol drug, we split the subject in two groups at random. Half the people get the new drug, half get a placebo. Nobody in the study knows whether they got the real treatment. And furthermore, even the people handing out the pills and otherwise dealing with subjects don't know. So that's to eliminate observer bias or confirmation bias, basically to eliminate bias. Okay, so that's some things about setting up the study. And now I want to talk about some things about the sampling. Bias when something in the way the experiment was conducted results in the treatment and control group differing in ways other than you intended, other than the way that you want to measure. So contrary to its use in everyday speech, bias does not necessarily imply that you did it on purpose. If you go to the Holiday Inn in Kansas City and you go through some conversation with people in the audience, somebody says, I think it might be that just because you arranged the study in the spring instead of the fall, you got that was entirely accidental, that nobody is saying that you cheated. So an example, the classic example, the example that we talked about a few minutes ago is voluntary response, when the sample members are self-selected. For example, a doctor testing a new drug asks patients whether they wish to try the new treatment or continue with the standard one. Another is that you put up a sign around campus that says something like, are you interested in quitting smoking? If so, we'll give you a free patch. People get patches for the shoulder. Who's showing up? Well, people who are interested in quitting smoking. So you're not getting the general population of smokers. You're instead getting a special subset. So here's some strategies for sampling that try to work out the bias. Randomizing, we've talked about it before, but it bears repeating. We use chance to put the experimental units, patients for example, into groups. The advantages are that you have smaller sample groups and you don't need to know the relevant factors in advance. The disadvantage is that because of the random choices, the experiment isn't repeatable, except repeatable on average. And we'll spend a good part of the middle of the semester talking about how you deal with the fact that the experiment is random and so in that way not deterministic. We can engineer around it, but we'll have to do that. So for example, you test the effect of aspirin on strokes by dividing the subject into three groups. First group takes no aspirin, second group takes 1 aspirin a day, the third group takes 2 aspirins a day. With randomization, the results will only be acceptable if they're statistically significant, if they can't plausibly be attributed to chance. We'll have much more to say about it later on in the course. But basically, if you find you have the three groups with no aspirin, a little aspirin, and a lot of aspirin, if you find a tremendous difference in the second group, okay, well now you have something that's significant. If you find, though, that the three groups are pretty much the same, some small difference, but pretty much the same, well, that's a different matter. Okay, matching. The experimenter tries to account for some of the influential factors. Studies of twins show that almost all traits are in part influenced by genetic differences. Some characteristics such as height showing a strong influence and some other characteristics a lesser degree. So some things have more complex hereditabilities with evidence for different genes affected different elements of the traits. So you might try to, for example, match people according to height. And your control group and treatment group are matched up in some way. Some vocabulary, kind of a lot of vocabulary, but this is the right place to put it, so I'm going to put it here. There are a variety of techniques that may suit a particular study. So far, we've talked about treatment group and control group mostly, but it can get more complicated than that. In a randomized experiment or a randomized comparative experiment, we assign values to the explanatory variable at random. We've talked about this a half dozen times already. And then you compare the response. The simplest thing is a simple random sample. Simple random sample, it gives every group the same size, it gives every group that size the same chance of being picked. So the classic example is we want to know about the new dining hall hours, so we go into the registrar's computer and pick 30 undergraduates at random, 30 students at random. Any size 30 group has the same chance of being picked. That's called a simple random sample because it's simple and it's random. In a probability sampling design, you deliberately adjust the chance that some members of the population are sampled. And the classic example, I hate to say it, it really drives me crazy, but the classic example is that retired people vote twice as often as 20-year-olds. Especially, especially this year, you hate to say that kind of stuff, but anyway, it's these days, but yeah, but it's true. So we take the sample in such a way that each retired person has twice the chance of being picked as each 20-year-old. So we've adjusted the probabilities. It's not simple. We've adjusted the probabilities. In a stratified random sample, you divide the population into groups and then pick randomly within each group. And again, this is a classic example here. You want to test a new weight loss pill and you solicit 100 volunteers, and who shows up for a weight loss study? Come on, it's mostly women. It's just a fact of our culture. Anyway, it's true. So maybe you get 80 women and 20 men. You're afraid that if you just take the entire group and split them using coin flip to put them into treatment and control group, you're afraid that just by accident you'll have 15 men and 5 men. So instead what you do is you take the men, split them 50-50 into the two, and take the women and split them 50-50 into the two. And that way your treatment and control group are sure to be balanced according to the sex ratio of the patients. In a matched pairs experiment, we give each case both treatments in some random order, or the cases are paired in some natural way. Husband and wife, let's say. But here's a good example. So we test a smoking cessation patch by getting 100 volunteers, splitting them at random into two groups. The first group gets the patch for 2 months, and then they get the patch with half dose for 2 months. The second group, we switch that around. We first give them the patch for half dose, and then the patch for full dose. And then we wait 6 months and we see which group had better luck with quitting smoking. So we matched. This patient got full and then half. These patients got half and then full. And finally, the most complicated one that we're going to think about, a multi-stage sample performs a selection in a series of stages. So the Australian Bureau of Statistics begins by dividing metropolitan regions into collection districts and selecting some of these at random. So they might look at a city and say, okay, we have 5 different regions in the city. We're going to select the Northwest and the Central East. Those are the 2 we're taking. We pick those 2 of the 5 at random. And then these districts are divided into blocks. So they take the Northwest and divide it into blocks, the Central East and divide it into blocks, and pick 5 blocks at random, 5 blocks at random. And then finally, within each block, you pick some houses at random. So when you decide to do the Northeast, that's it. You're not doing Northwest. You decided to do 2 of the 5 regions. You're not doing the other regions at all. They're gone. So in a multi-stage sample, we draw a random sample from, in a stratified sample, we draw a random sample from all the strata, whereas in a multi-stage or cluster sampling, we study only selected clusters. You're just trying to reduce the number of people that the Australian Bureau of Statistics has to approach and ask questions. So that's a relatively complicated approach. One case that people run across a lot in kind of everyday life, you read about it in in the newspaper or online, and so it's something that people often see. So I want to talk to that point specifically, and that is sampling. You go up to a bunch of people and ask them questions. Here are 4 things that can give trouble in a sample, 4 things to be mindful of in a sample. First one, undercoverage. Some part of the population may just not be represented. Just not represented. Okay, so for example, Star Trek. Star Trek was on TV when I was a kid. Star Trek is a tremendous, been around for tremendous success, been around for decades and decades, made tons of money for somebody. Star Trek was, in fact, when it first appeared on television, was a failure. It was only on for 3 seasons. It never did well. Why? How's that possible? And the reason is, of course, that Star Trek was not successful with the people who were being measured. The people who were being measured, the people who the television rating company was measuring, was not college students. They just threw up their hands and said, we can't measure who college students are watching. Who watches Star Trek? Well, it's that kind of age. So the company was not collecting data on who was actually watching the show. So that's why it was never a commercial success when it first came out. Non-response or voluntary response. Some subjects may fail to respond. Some subjects may over-respond. We've talked before about if we ask St. Michael students for hours in the dining hall. What hours would you prefer? We would get very, very few responses. So we have a tremendous difficulty with non-response. And the people who do respond, you say to yourself, are probably not typical. There's nothing wrong with asking their opinion, but they're probably not typical of the wider student body. Some subjects may fail to respond or may over-respond, and the example I have there is that in a recent Vermont governor's race, the paper incorrectly predicted a Democratic victory, obviously the Republican one. The explanation was that they did phone polling, and more Republicans had phone answering machines, and so more often Republicans were non-respondents, and so they had a problem with who was in their sample. Response bias. Sometimes something in the behavior or appearance of the questioner affects the responses. People will respond differently to questions about domestic violence if the questioner is a male than if the questioner is a female, or if the questioner is wearing a tie, this kind of stuff. It makes a difference. It makes a big difference. And finally, the last one here, the wording of questions. Small changes in the wording may greatly affect the responses. I mean, I gave a dramatic example. Asking it, since 18-year-olds are old enough to fight for their country, should they be able to drink alcohol, is obviously encourages certain response. But you will get a different response if you sample people by saying, what's your opinion of the president, 1 to 10, versus what's your opinion of, and name the president. You'll get different responses. If you hire a company to do a survey for you, most of the time you interact with the company. They'll be asking you about, well, this wording, that wording, kind of. You're trying to tweak it to get the most accurate responses. Okay. Another lesson about sampling is that randomization is very hard. I remember when I was a kid in 1969, the U.S. held a lottery to determine who would be drafted in the Vietnam War. And they wrote all the birthdays on basically like ping pong balls, and they put them in a big tub, and they mixed it up. And then the Secretary of Defense reaches into the tub, and he pulls out the first number. And if that was your birthday, you were going. People pretty soon noticed that December birthdays had more chance of being drafted than January birthdays. It didn't take long for people to figure that out. That is to say, although they thought they mixed up all the balls, they didn't. Randomization is a lot harder than you think. Today, professionals will generate the random numbers on a computer, which can be tricky itself, but nonetheless is doable, or else use a table of random numbers. You can see that that's a link, so you could click on that and go to Amazon to buy a table of random numbers. So the summary. So I hope that I've convinced you that although we all grow up in a Newtonian culture, we all grow up thinking that the world is continuous, that a small change in the way you do things will cause a small change in the outcome, that in fact some things in life are not that way, and sampling is an example. Statistical studies are an example. You know, I think we've all had the experience of, let's say, you go to the post office, you got 3 packages for Aunt Joan. You put them on, the 3 of them on the counter, and the 3 of them get put into the box to be taken away. And for some reason that you can't figure out, the 3 packages don't arrive at Aunt Joan's on the same day. A small change in the way they were handled, one of them got on this truck, one of them got on that truck. A small change in the way they were handled caused a large change in what day they arrived. Well, statistical studies are the same thing. A small change in the way you lay the study out, historical controls, can completely reverse the conclusion. So here's an example. You want to test if elementary school students learn math better by watching videos. It's convenient to break it up into boys in this room, girls in that room. It may be convenient, but when you get to that holiday in Kansas City, somebody's going to raise their hand and say, but there's a difference between treatment and control group besides whether they watch the videos or not, and the difference in this case is the sex of the subjects. Now please, let me close with, life is complicated. You can't ethically settle the question of whether exercise prevents people from getting dementia by grabbing 100 babies and forcing 50 of them to exercise all the time for the next 80 years and forcing the other 50 to sit around the house for the next 80 years. Nobody is saying you can do such a thing. No. But to the extent possible, when you make it your study an experiment rather than an observation, to the extent possible, you make it better. You settle more questions that people will be asking about your study. Even if you came to the right conclusion, it may still be an unconvincing study. To the extent possible, you want to make the study an experiment. Okay. All right, very good. Long talk today, but I'm trying to make, I hope, trying to make a key point about when you look at a study, the way you lay the study out is item number one. Was the study laid out in the best possible, practical, ethical way? Investigating the study, analyzing the numbers, all that kind of stuff. We're going to talk about that at length, of course. But item number one is, was the study well done? Okay. All right. Very good. See you next time.